All right, let's read chapter 2 after our quick introduction of Children of the Grave. This is Whistling Past the Graveyard. We're going to read it. No commentary, no narration. I may do a follow-on video for chapter 1 with additional narration. Some of this chapter was started yesterday in episode 1 of this 286 series, which is a redo of a reading of this book, which I did a few years ago. Paul Luciu was alone in his lab at Chiron Corporation in Emeryville, California on an August evening in 1984. Rush hour traffic on the nearby Nimitz Freeway had fallen to a dull rumble. The technicians had gone home, and Paul Luciu relished the chance to work without interruptions. Again, Luciu is Luciw. Then the call came from Kathy Steimer, last name Steimer, his closest colleague at the company. Paul, I've got a problem, she said. The ultracentrifuge has stopped. He could picture exactly where she was, all alone in a spooky P3 containment facility, a place where dangerous microbes could be handled in an isolated, specially ventilated room. This lab, a part of the Navy Biosciences Laboratory, or NBL, was less than 2 miles from Chiron. All summer, Paul Luciu and Kathy Steimer, Paul and Kathy, had been loading the huge, heavy ultracentrifuge with samples of virus the doctors had isolated from AIDS patients in San Francisco. Like other teams of scientists in the United States and France, they were racing to unravel the genetic code of the virus and figure out how this tiny killer is organized. Scientists put a premium on this information because it could open the door to diagnostic tests, treatments, or even a vaccine. When Kathy called, Paul's first thought was that the ultracentrifuge had thumped and bumped itself off balance like an overloaded washing machine, wreaking havoc with its contents while whirling at up to 20,000 revolutions per minute. If this had happened, whoever lifted the lid would find a jumble of metal racks and broken glassware coated with deadly viral goo. Fortunately, Kathy reassured him, the situation wasn't as serious as that. The ultracentrifuge had simply ground to a halt while she was running a large preparation of virus. What troubled her was the prospect of filing an incident report required anytime something goes awry in a lab. With the central NBL office, and again, that's the Navy Biosciences Laboratory, the NBL, with the central NBL office. Kathy and Paul had been cleared to work in the lab only because a Navy researcher, who had happened to be an old friend of Chiron's CEO, I don't know who that person is at this time, had lent surplus lab space to the company 3 months earlier. This made it possible for the fledgling biotech to work on the AIDS virus. At this point, the company hadn't finished building a place where dangerous organisms could be safely handled. As guests of a military facility, working on a virus that terrified most people, the Chiron scientists had kept their heads down. As soon as Kathy finished talking to Paul, who agreed that they had to reveal what microbe was in the broken centrifuge, she called the duty officer for the biosciences lab. He immediately notified the commanding officer of the Oakland Naval Depot, where the laboratory was located, to report that there had been an accident with the AIDS virus on the base. The irate commander demanded to see Kathy Steimer, in his office that night, not in the morning, but ASAP, as soon as possible. If the base commander was so bent out of shape, what would happen to the viral cultures that she had spent all summer cajoling into lush growth? For all she knew, a hazardous waste disposal team was already hurtling in her direction, with instructions to confiscate the precious flasks. In a second quick phone consult, Kathy and Paul agreed that it made no sense to wait and see. Within minutes, Paul was on his bike, pedaling furiously from Chiron's headquarters in Emeryville, California, a down-at-the-heels industrial suburb of Oakland, to the Navy base. Paul was a short man, only a few inches over 5 feet tall, but fit in the way of one who shuns cars and walks or bikes wherever he goes. Once at the Naval Depot, Paul showed his security pass to the armed guards at the gates and pedaled on to the P3 building, a forlorn structure near the base's edge. In the entry chamber, he stripped off his jeans and T-shirt and tossed them into a locker, pulling on a cotton scrub suit, gown, masks, booties, and latex gloves. As soon as Paul stepped into the ill-lit hallway, lined with derelict freezers and obsolete lab equipment, he began to sweat. His reaction was the same every time he saw the labels on the closed doors of individual labs. The hallway was a rogues gallery of pestilence, plague, anthrax, slow viruses that eat the brain. God knows what scourges had been handled in this old building and by whom. The only room in use now, however, was the one where he and Kathy had labored 14 hours a day, 6 or 7 days a week since May 1984. All the time, they had never seen another person in the corridor. Kathy had removed the contents of the ultracentrifuge and had lined up the T-150 flasks on the bench. Each contained about 1 cup of cloudy viral broth, and she was tightening the tops and wrapping them in tape. Working swiftly, the two identically dressed scientists put each flask into a plastic bag, sealed it, then bagged it again. Finally, they loaded the flasks into some of the thick styrofoam shipping boxes that accumulate in biology labs like coat hangers in a closet. These they sealed with broad strips of plastic shipping tape. The goal was to move the cultures to the small biosafety level 3 room that Chiron had completed only a few weeks earlier. Clearly, they'd have to transport the boxes of virus in Kathy Steimer's car, but leaving through the front gate would be too risky. It was now late in the evening, and the guards would surely suspect these civilians of stealing government property. A stealthier departure would be better. Soon, Kathy traded her sterile garb for street clothes, retrieved her car from the nearby lot, and signed out at the guardhouse. A couple of quick turns put her on a service road that hugged the outside of the Navy's tall chain-linked fence topped with concertina wire. She rolled slowly through the bright pools of the tall security lights, finally dousing the headlights and stopping in a dark patch only 20 yards from the back door of the lab building. She slipped out of the car, taking care not to slam the door. Paul had been watching, and within seconds he emerged from the lab, looking nervously for the armed sentries who patrolled the base at night. He listened for the crunch of tires, but heard only the low whiz of light traffic on the freeway. The air was brackish with the smell of the nearby mudflats of San Francisco Bay. With a box tucked under each arm, he hurried back and forth from the door to the fence until all the shipping containers were out. Kathy and Paul each took a few steps back from the fence. She was the taller of the two by half a foot, a rangy, flaxen-haired California girl. And for those who are curious, I actually had to look it up. I'm not ashamed to say it because I learned things. Flaxen means golden. So she had a rangy, golden-haired California look. He was smaller and darker, and his spectacles glinted in the darkness. They exchanged furtive whispers and a signal nod before Paul gently lofted the first box, like a square and fragile beach ball, over the fence to Kathy's waiting arms. One by one, she received them. Less than an hour later, all of Kathy's precious flasks filled with the AIDS virus were safe on the Chiron campus. She returned to the Oakland Navy Depot alone to meet with the base commander. He was not a scientist, meaning the Oakland Navy Depot commander, so Kathy explained calmly and carefully that although a machine had malfunctioned, no deadly organisms had been spilled and nothing had been contaminated. She also explained that she and Paul had been authorized to use the containment facility by the Navy scientist in charge of it. The commander did not care what had happened or how it came to be. Only the future interested him. The AIDS virus, he told Kathy, was not the kind of problem he was going to have on his base. In 1981, the same year that doctors in Los Angeles and San Francisco began reporting puzzling and rapidly fatal illnesses among young gay men, the fledgling Chiron Corporation made history by inventing a new kind of vaccine against hepatitis B. Some cases of this viral infection were chronic, untreatable, and ultimately fatal. And like AIDS, it was commonplace among homosexual men and posed a threat to transfusion recipients and healthcare workers who handled human blood. Chiron's founders, a man named William Rutter, Rutter, and another man named Edward Penhoet, Penhoet, were academic biochemists at the University of California campuses in San Francisco and Berkeley, respectively. Both were experts on hepatitis B and other viruses that prey on people. More importantly, they were charter members of a small club of genetic engineers. They practiced recombinant DNA technology, which basically meant snipping genetic material out of one organism, inserting it into a different organism, and tricking the host into expressing it, churning out the protein specified by the newly transferred DNA. This enabled them to make a large amount of whatever protein they choose, which might hold promise as a preventive vaccine, a treatment, or laboratory tool. On Easter Sunday of the year 1981, Rutter and Penhoet wrote out a wish list of products that their new company might make using recombinant DNA technology. Their goal was not only to solve some real biological mysteries, but also to use genetic engineering to improve human health. At the top of their list were vaccines to prevent hepatitis B, herpes, and influenza, as well as tests that could be used to check blood samples for two types of hepatitis, B, and the mysterious newer kind known as non-A, non-B. Since then, it's been shown to be several distinct viruses, including hepatitis C. Rutter's lab at UC, or University of California, San Francisco, had already studied the hepatitis B virus in great detail. At the time Chiron was founded, the East Coast pharmaceutical giant Merck was actively seeking a replacement for its current hepatitis B vaccine, which used a protein derived from the blood of hepatitis B carriers. Making vaccine this way was not only expensive, but the process was also vulnerable to contamination with other blood-borne viruses, including a new scary pathogen responsible for AIDS. Merck wanted a new kind of vaccine that would cost less to make, could be manufactured in a laboratory, and would be free of contamination. This was a tall order, however, and the best ideas for such a product were being nurtured in young biotech companies or in academic labs. Merck invested in several groups racing for the same goal, and one of them was Chiron. Chiron's main rivals were hotshot researchers at Genentech, that's Genentech, the San Francisco Bay Area's preeminent biotechnology company, and a group of Rutter's former collaborators at the University of Washington in Seattle. But it was Chiron's Pablo Valenzuela who got there first. He figured out how to use recombinant DNA technology to make the hepatitis B vaccine of Merck's dreams. From hepatitis B virus, Pablo Valenzuela removed DNA that coded for a protein on the outside of the virus called HBsAg, short for hepatitis B surface antigen. Spike protein. Spike protein. He then inserted the DNA into yeast cells, which were cheap and easy to grow, and viola, the yeast cells cranked out HBsAg that looked like the protein Merck had been laboriously isolating from blood. This genetically engineered version could be made safely without any blood or virus involved. By Christmas, Tiny Chiron had outdistanced its competitors and licensed this product to Merck. This precedent-setting vaccine, approved for sale by the FDA, Food and Drug Administration, in the year 1986, does an excellent job of preventing hepatitis B and is used around the world. All vaccines accomplish the same thing. Like a self-defense instructor impersonating a mugger, the vaccine teaches the immune system what to do in an emergency. When a real thug appears, the lesson is recalled, and a swift immune response immediately overpowers the attacker. Most traditional vaccines consist of disease-producing organisms that have been altered or killed. The Sabine polio vaccine is made from live attenuated or weakened virus, for example, whereas the Salk formulation consists of killed organisms. Sometimes nature supplies a vaccine. Credit for eradicating smallpox goes to vaccinia, a pox virus that originally infected cows, but in humans evokes a protective immune response to smallpox without causing any harm. Today, recombinant DNA technology offers a wondrous alternative, a wholly new generation of vaccines that are safer and cheaper to make than old-fashioned ones. The first step towards building a genetically engineered vaccine is to identify an antigen that is always part of the attacking virus. Antigens are simply molecules, usually proteins, that the immune system recognizes as a threat and takes steps to eliminate. Finding the right antigen, as it turns out, can be an enormous challenge. Pablo Valenzuela's hepatitis B vaccine was a cosmic home run for Chiron. Suddenly, anything seemed possible. In order to keep the company's streak going, Bill Rutter and Ed Penhote drew up lists of scientists they wanted to hire, and most said yes when they were asked. Penhote recalls, quote, "We were looking for people with a high energy level, dedicated to their work, and who wanted to make a difference." In 1981, the scientists who joined Chiron and other biotech startups were turning their backs on stodgy academic tradition and offending many of their mentors. During the late 1970s, the world of biology was split over the legitimacy of patenting discoveries made in the course of tax-supported research. Old guard academic scientists saw university-based research as a gentleman's game and looked down their noses at scientists who went into trade by joining a biotechnology company. Today, when professors at elite universities and medical schools race to patent discoveries as soon as they are made, this snobbishness seems as outmoded as the idea that all telephones must have cords. But this attitude was entirely real in the year 1981. Penhote recalls, quote, "So we put people on our list that we thought we could actually recruit, and they were all young people," unquote. Virologist Paul Lushu was 33 when he joined Chiron Corporation in July of 1982. He had just completed postdoctoral training in a UCSF, University of California, San Francisco lab, whose leaders would win the Nobel Prize years later. Rutter, Penhote, and Valenzuela were a tremendous lure for Paul Lushu. Not only were they respected as top-notch scientists, but, quote, "they were adventurous, broad in their thinking, yet able to define goals and get things done," unquote. Hiring Paul was a smart move for Chiron because he was an expert on retroviruses, bizarre little organisms that were just beginning to seem relevant to human health. Although doctors weren't worried about retroviruses in the pre-AIDS era, veterinarians were all too familiar with the damage these unusual organisms caused. Feline leukemia virus, known as FELV, caused many thousands of fatal cancers in domestic cats. There would be a lucrative market for a vaccine against FELV, and this was Paul's first project at Chiron. He explored two possible approaches at once: One was a classic vaccine strategy in which protection would come from immunization with a weakened version of FELV itself. The other, and far more exciting, idea would be to mimic the recombinant hepatitis B vaccine, splicing FELV genes into yeast cells so that these cells turn into vaccine factories. Kathy Steimer had just turned 34 when she decided to write to Pablo Valenzuela and ask for a job at Chiron. She and Martin Wilson, a young neuroscience professor she had been dating during the previous year, drafted the letter beside a swimming pool in Davis, California. They had been introduced the previous Christmas by a mutual friend and had become inseparable. Their lives had begun quite differently, with Kathy growing up as the daughter of a firefighter in Los Angeles, and Martin grew up in a cultured British household where he was immersed in classical music. Both families valued education, however, and each produced a scientist absorbed in the challenges, frustrations, and satisfactions of laboratory research. In addition to falling in love, Kathy Steimer, and Martin gave each other tremendous professional support. So it was natural for Martin to help her write a job-hunting letter to Chiron. Like Paul, Kathy Steimer was a skilled retrovirologist. Earlier, she had left a biotech company because she was impatient with the managerial incompetence, then moved to an immunology postdoc at Stanford, where she grew tired of being patronized by male physicians who were sure they were smarter than any woman with a PhD. At this point in her career, Kathy wanted to be respected for who she was and what she knew. During her first biotech job, she'd worked next door to Chiron and had seen Rudder, Penhote, and Valenzuela in action. Observation told her that they were the kind of people she wanted as colleagues. The feeling was mutual, and Chiron needed a scientist with exactly Kathy's strengths. An earlier postdoctoral stint at a famous Harvard lab specializing in growth factors, natural substances that signal cells to grow or divide, had made her an artist when it came to growing temperamental cells. At Stanford, which had not been a happy experience on a personal level, Kathy had worked on the cutting edge of immunology. In Chiron's view, she was exquisitely prepared to create new assays for determining how the human immune system responds to experimental vaccines. Kathy Steimer started at Chiron Corporation in September of 1983. She promptly devised several methods for detecting viruses that proved useful for research purposes, had less success with a crotchety system for automating certain lab tests, and figured out how to synthesize interleukin-2, usually called IL-2, a cell growth factor that was fun to tinker with in the laboratory but didn't appear to have any immediate medical value. 2 months after Kathy started to work at Chiron, a 31-year-old molecular biologist named Nancy Hagewood. And this is one of the main reasons why this book is valuable, came to the company from another, less successful biotech in Maryland. At this point, there were biotech startups on every corner, but in Nancy's mind, Chiron and Genentech were the only two destined for success. A military brat, Nancy Hagewood grew up on overseas bases where cholera, yellow fever, and the like were commonplace. These early experiences sparked her interest in biology and in human disease in particular. She joined a biotech company immediately after her postdoc at Johns Hopkins, feeling that the private sector was the best place to be if you wanted to develop products that helped people. On that first job, Nancy Hagewood met and married a protein chemist who also joined Chiron when she did. Nancy was ambitious and, in her own words, quote, "I wanted to work in a really high-powered place because I thought it would be fun," unquote. She brought important skills to the company. She was an expert at site-directed mutagenesis, a technique for introducing deliberate mutations into a gene. Another major interest was splicing genes into animal cells so that they could be turned into protein factories. Although Chiron made its successful hepatitis B vaccine in yeast, some scientists believe that future vaccines should be made in mammalian cells that more closely resemble human cells. The first generation of molecular biologists thought there was no disease that they couldn't conquer, and their dreams were paid for by investors besotted with biotechnology. Dozens of other bright young scientists cast their lot with Chiron in the early 1980s, and they were backed by executives and investors brave enough to risk the time, money, and space needed to tackle major medical puzzles. But it was mainly the brains and determination of Paul Lushu, Kathy Steimer, and Nancy Hagwood that enabled the company of Chiron Corporation, to create one of the first experimental vaccines for AIDS. The New York Times wrote in November of 1983 that AIDS cases had been identified in 33 countries and on every inhabited continent. By Christmas that year, epidemiologists at UC San Francisco estimated that 1 of every 3 gay men in the city was already infected with the virus that causes AIDS. San Francisco was hit early and hard by the new epidemic, and although cases had been identified among hemophiliacs, transfusion recipients, IV drug users, and a few prostitutes, most people saw it mainly as a gay disease. AIDS was an ugly way to die. The virus decimated the immune system, leaving the patient vulnerable to opportunistic infections by microbes that aren't normally a problem for healthy people. AIDS patients were disfigured by the purple lesions of Kaposi's sarcoma, wasted by intractable diarrhea, and suffocated by a vicious pneumonia. Microbes swarmed into every orifice. Mouths turned mossy with yeast infections, and bowels were lacerated by bacteria. Some patients went mad with dementia as malignant lesions swelled in their brains. Doctors prescribed antimicrobial drugs for opportunistic infections, but this was like trying to halt a locomotive with bare hands. Rudder and Penhote and their scientists knew all about this. Part of Chiron's business strategy was to maintain close working relationships with UC San Francisco and UC, again, University of California, Berkeley, where they still had strong ties. The human catastrophe of AIDS was all too evident at UCSF and its crosstown teaching hospital, San Francisco General, which was on the front line of the epidemic. Many of the previously healthy young men who were dying of AIDS were the first people who had earlier been infected with hepatitis B. And this, after all, was the disease that had already made Chiron famous. Although it would be romantic to think that scientists throw themselves at a new infectious scourge because it vexes their curiosity or stirs pity and terror in their hearts, this isn't how it works. In fact, researchers tackle problems that they believe they are equipped to solve. Rudder and Penhote figured that they had at least some of the tools needed to study whatever virus might be causing AIDS. At the very least, Chiron might come up with a blood test that could confirm the diagnosis in patients or screen donated blood for signs of the virus. The company already had a test for detecting hepatitis B in donor blood, and if it also had a similar one for AIDS, it could package the two and sell the product to blood banks. More important, detailed knowledge of the hepatitis B virus was what enabled Chiron to make the world's first genetically engineered vaccine. If Chiron put its best scientists to work on the AIDS virus, who could say? Back-to-back homers are not unheard of. In 1983, the French researcher Luc Montagnier and Robert Gallo of the National Cancer Institute each reported that he had isolated retroviruses from patients sick with AIDS. Luc Montagnier called his virus LAV for lymphadenopathy-associated virus. Doctors had noticed that many of their gay male patients suffered lymphadenopathy, chronically swollen lymph nodes, before they developed symptoms of AIDS. Montagnier isolated his virus from some of these patients' lymphoid tissue. Gallo dubbed his virus HTLV3 because he believed it was the third in a family of what he called human T-cell leukemia viruses. Hard on her heels was UCSF virologist Jay Levy, last name Levy, who had also isolated a retrovirus from AIDS patients in San Francisco. He called his ARV for AIDS-associated retrovirus. Were these three viruses the same? Were there three different viruses on the loose? Nobody knew. Nor was it clear whether any of them actually caused AIDS, or whether they were among the pathogens that overwhelmed patients left defenseless by immune system collapse. These were tough questions that Levy needed collaborators to answer. Not only that, but if the answers were going to make a difference, he needed ties to a company that could convert new findings into clinically useful products. In April of 1984, Jay Levy began sending Chiron samples of virus that he had collected from AIDS patients. In addition to the impeccable UCSF pedigrees of Chiron's top guns, this small biotech had three bona fide retrovirologists among its several dozen employees. Dino Dina, first name D-I-N-O, last name D-I-N-A, was the head of the vaccine division, Paul Luciu and Kathy Steimer. Their expertise was crucial because retroviruses are not like other living things or even viruses. Narrator's note, I believe if you go to the archive, I don't have a lot on Dino Dina. There's a picture of him. I've a copy of some of the papers that he's worked on. Here is one bio. Dino Dina served as the head of Dynavax Technologies Corporation from May of 1998 to 2013. He was also president of a company called Dynavax. He joined Dynavax in 1997. Dino Dina was an employee of Chiron Corporation, a biopharmaceutical company. At Chiron, Dr. Dina held a series of positions with increasing responsibility. He ultimately served as president at Chiron Vaccines, formerly Biocene Company, which he directed from its inception to 1984. Under his direction, Chiron's vaccines received the first ever approval for an adjuvanted influenza vaccine in Italy, successfully completed the development of the first genetically engineered pertussis vaccine, and conducted clinical trials for vaccines to prevent HIV, herpes simplex type 2, cytomegalovirus, and hepatitis B infections. There are many scientists mentioned in this book, Big Shot AIDS Vaccine, published the week after September 11th, 2001, by Patricia Thomas, that are hard to get solid bios on. Sometimes it's because there wasn't much written about them otherwise. Sometimes it's because it's been censored. Anyways, I have as much as I can on many of the people. This work was done years ago, and it's still available. It's still alphabetized, etc. Getting back to the book, Dino Dina was the head of the vaccine division, Paul Luciu and Kathy Steimer at Chiron. Their expertise was crucial because retroviruses are not like other living things, or even other viruses. Most life forms store their genetic code as DNA. When a plant or animal cell divides, its DNA is transcribed into RNA, which in turn tells parts of the cell how to make proteins. These proteins enable living organisms to go about their business. If the human body were a large hotel, proteins would be its brick and mortar, its plumbing system and telephone wiring, the furnace in the basement, and the stove in the kitchen, even the housekeeping staff that keeps order and hauls out the trash. The DNA packaged into the nucleus of every cell is the master plan for building and running the hotel. Different types of cells follow different parts of the plan, communicated to them in the form of RNA, and like subcontractors, they follow their specific set of instructions. Some build walls, some install communication systems, others move raw materials from place to place. Each separate part of these jobs is a protein. All right. The DNA makes RNA makes protein pattern also holds true for most viruses. What sets viruses apart from other organisms, however, is that they can't manufacture proteins on their own. So, in order to accomplish this, their DNA must invade another creature's cells and commandeer its subcontractors to churn out more virus. Retroviruses have an even stranger way of perpetuating themselves because retroviruses use RNA as their genetic blueprint instead of DNA. Because retroviruses use RNA as their genetic blueprint instead of DNA, they require an extra step not needed by other viruses. A retrovirus uses an enzyme called a reverse transcriptase to translate its RNA into DNA. That DNA slips into the genetic code of the infected cell, where it is turned into RNA instructions that direct the cell to make proteins the virus need to reproduce. The first hint that retroviruses might be medically important came in the early 1970s, when one of these contrarian viruses was discovered to cause cancer in chickens. Links to leukemia in cats, cows, and other animals soon followed. While it made sense to suspect that retroviruses might also cause cancer in humans, this connection was so difficult to uncover that tired, increasingly cynical researchers called them, quote, human rumor viruses, unquote. The jokes were silenced in 1980 when Gallo demonstrated that a retrovirus could cause human T-cell leukemia. Soon afterward, he linked a related virus to a hairy cell leukemia. Gallo called these retroviruses HTLV1 and HTLV2. Young retrovirologists like Kathy and Paul grew up with these discoveries and couldn't wait to get their hands on the AIDS virus. The leukemias caused by HTLV1 and 2 claimed a few hundred lives each year. The human toll of AIDS was far greater. In 1980, it had been unknown, but by 1984, there were more than 200,000 infections in the United States and more than 1 million worldwide. Fears about personal risk took a back seat to their overwhelming sense that this was the biggest challenge of their careers. The stakes were high for Kathy Steimer and Paul Lushu, and unlike Gallo and other established scientists who now spent most of their time at desks, these passionate young researchers were willing to spend long hours at the lab bench, which is exactly what Chiron wanted them to do. Bill Rutter, in particular, felt that it was important for Chiron to analyze the virus and generate data that could be patented, paving the way to diagnostic tests and vaccines that Chiron would own. Down the road, when detailed knowledge of the virus had been transformed into marketable products, Rutter wanted his company to be collecting royalties, not paying them to somebody else. Although Chiron had skilled virologists, it didn't have a safe place to work with anything as lethal as the AIDS virus. Fortunately, the company was able to borrow an unused Navy P3 lab, which had the isolation barriers and special ventilation needed to contain dangerous pathogens. The Chiron team brought in some new equipment and supplies, turning the old lab into command central for the new AIDS project. They set to work on samples of the virus that Jay Levy called ARV, collected from patients in San Francisco and driven across the Bay Bridge in sealed containers. By May, Kathy Steimer and Paul Lushu had made an important scientific decision of their own. they decided to study patient samples from a second source, the Medical Pathology Department at the University of California, Davis, located 90 minutes away in Sacramento. Although Levy was certain that his virus was indeed the cause of AIDS, Kathy and Paul had been taught to assume nothing. If they studied the virus only in the same group of patients where it had originally been found, they would know only that it occurred in those patients. More patient samples obtained from an independent source in a different locale would be essential if they were going to make judgments about the true importance of ARV. The more places it turned up, the more likely it was to be the true cause of the disease. Paul Lushu's most important job was to clone the virus's entire genetic code and to determine the exact sequence of the chemical letters that make up that code. Outside of science fiction, most people had never heard of cloning until Dolly the sheep made her debut in the year 1996. Actually, the term has a longer history and broader meaning than does the recent interest in copying farm animals. In molecular biology, clones are genetically identical copies of a gene, a cell, or an organism. Cloning a viral genome, meaning its entire genetic code, involves cutting the genome into pieces and inserting those pieces into a biological system that churns out millions of identical copies. A virus that has been isolated from blood can't be patented. One that has been cloned and sequenced can be. That's why Paul's work was so important to Chiron. Kathy Steimer's project was also crucial because her task was to invent a test that could be used to screen human blood for antibodies to the AIDS virus. The government had already announced that an antibody test was its top priority for fighting the epidemic, and Chiron expected this to be its first AIDS-related commercial product. Check this out. This is a pretty hard book to find. HIV Detection by Genetic Engineering Methods. Edited by Paul Lushu, Kathy Sue Steimer. It's mostly a collection of papers. Publication date 1989. None of the names in here ring a bell. 1989, from a guy my age, it seems like it couldn't be that long ago. And then you start doing the math, and you're like, that's closing in on 40 years. Wow. That's, I'm telling you what, if you're in my audience and you're a young member, and I know I don't have many young members in my audience, unfortunately, even though I'll tell you what, it just goes by real fast. Scary. Anyways, Paul Lushu and Kathy Steimer are key people in this book. So, without getting into the narration, let's finish up the work here. The AIDS virus is the ultimate example of a creature that bites the hand that feeds it. It penetrates T4 lymphocytes, certain white blood cells that are essential for a healthy immune system. Once the virus is inside, it hijacks the T4 cell's machinery to make more of itself. By doing so, the AIDS virus damages and eventually destroys its host. Sometimes a T4 cell is killed when millions of virions, or baby viruses, explode its membrane like the skin of an overripe tomato. Other T4 cells expire less dramatically when their own metabolic machinery is choked to a halt by wads of viral genetic material and proteins. Not only did the AIDS virus's relentless destruction of T4 cells kill patients, but it was also a major headache for every scientist who tried to grow it. Whether the setting was Paris, the National Cancer Center, or Chiron's improvised lab on the Navy base, cultures died as the virus gobbled up all the cells in a closed test tube or flask. There was immense pressure to grow great quantities of virus as fast as possible, and the biologists held their secrets as closely as winemakers. Unlike the experiments that many people remember doing in high school or college, where success hinges on little more than the ability to follow directions, Kathy Steimer and Paul Luciu had no lab manual to follow. Swaddled in sterile garb from head to toe, they worked alone in the Navy isolation facility. Paul's cultures were in T75 flasks, each holding about 1/2 cup of material. Kathy worked in T150 flasks, twice that size. They moved slowly, carefully, like dancers, not wanting to break or spill anything, or even to breathe the air that was released when they pulled the stopper out of a flask. Kathy and I trusted each other, Paul would later say. He went on to say, if there had been more people in the lab, there would have been the element of mistrust, of wondering whether others were being as careful as you are. That adds a lot of tension. Neither Kathy nor Paul was willing to expose Chiron lab technicians to such a dangerous microbe. In the year 1984, it was clear that the AIDS virus could be transmitted by blood, but virologists didn't know whether it might also spread by aerosols, tiny infectious particles that could be breathed into the lungs. Not only that, but how could the scientists tell technicians what to do when they were improvising every move? Kathy and Paul fed the cultures with biologic broths that they hoped would nourish the T cells and keep the virus growing. They added dribs and drabs of this and that, chemicals called growth factors, which stimulate cells, and chemicals called cytokines, which cells use to communicate among themselves. They used different T-cell lines. Some they'd purchased from a biological supply company, some they'd gotten from J. Levy. For more than a month, they couldn't get a culture established no matter what they did. When they talked, it was about what to try next. Finally, in early June, 1984, presumably, all their conscientious tinkering paid off. The virus grew happily in a T-cell line that had come from J. Levy. Now, Kathy Steimer and Paul Luciu were excited and spent more time in the lab than ever. Martin Wilson, who later became Kathy's husband, remembers that a typical Saturday night date during this period involved going with Kathy to the eerily deserted Navy containment facility, suiting up in scrubs, and watching Kathy pour supernatant, the liquid from the flasks, into containers that she spun in the centrifuge. Martin said, quote, "It scared the shit out of me. I always thought, give me one little bit of aerosol here, and who knows what can happen." On the small television in the corner of the Navy lab, the games of the 23rd Olympiad played out as Kathy and Paul cultivated increasing quantities of the AIDS virus and began experimenting with it. Most of the time, they were intensely busy, but sometimes they were forced to wait while the centrifuge spun or the electrophoresis gels worked their magic. In these small boxes of translucent gelatin, droplets of liquid were lined up like sprinters in the starting blocks. Drawn from one end of the box to the other by the invisible pull of a tiny electrical current, the distance traveled by each sample told the researchers exactly what it was. These little races were slow and painstaking and had to be run again and again and again and again to make sure the results were right. Each time they looked up to see Carl Lewis winning yet another gold medal, it was a reminder that sports was gloriously simple compared to science. Or at least it was faster. The pressure to make progress against AIDS was intense in 1984. Rudder and Penhote could stand in their offices on the third floor of 4560 Horton Street in Emeryville, California, and see San Francisco's skyline across the bay. The disease was rampaging through the city's gay community, and there was a huge brouhaha about shutting down the bathhouses where men went for anonymous sex. In Atlanta, the United States Centers for Disease Control, the CDC, announced that most of the nation's hemophiliacs were already infected with the virus and that the number of cases related to blood transfusions had quadrupled in 4 months. There was widespread panic about AIDS and donated blood, and the National Institutes of Health, or the NIH, was ready to pump money into tests that blood banks could then use to protect the general population, an unsubtle way of saying heterosexuals who didn't inject things. Although Kathy and Paul had worked incredibly hard to grow the virus, when they returned to Chiron headquarters, the pace was so intense that their time at the Navy P-3 facility seemed tranquil by comparison. The ultracentrifuge accident ended their stay in the deserted Naval Biosciences Laboratory, and now they were buffeted by noise and distractions on all sides. Chiron was housed in an old research building that had been used by Shell Oil, then abandoned in Emeryville's industrial flatlands until Chiron had dusted it off and reopened it in 1982. On one side of the long third-floor corridor were Chiron's executive and administrative offices. On the other side was a row of large, open laboratories, facing not the bay, but the Berkeley Hills. There was no air conditioning or heating in the labs, and on hot days, when the tall windows were open to catch a breeze, the air was heavy with the stench of a nearby plant where cattle were rendered into hides, fertilizer, and the like. Grit and pollution from the busy freeway feeding the Bay Bridge settled on the lab benches and had to be constantly wiped away. The PhD-level scientists and their lab technicians, many of whom were fresh out of college, wore jeans and listened to the radio while they worked. What set the techs apart was that they worked only 8 hours a day. Paul lived in a large condominium complex on the edge of San Francisco Bay, a short walk or bike ride away from Chiron. He didn't have a phone in his condo because he was rarely there. Paul usually started work around 10 or 11 a.m., and left around 2 the following morning. Kathy and Paul shared a small office next to the lab, and sometimes she would come in at her usual starting time around 9 a.m. and find him asleep on the office floor. Nancy Hagewood's lab was just down the hall, and although she wasn't working on the AIDS project yet, like all Chiron scientists, she was constantly in overdrive. She said, "We worked all the time. It was rare to take a whole weekend off. We worked really hard because the company's survival depended on us, and we were really into what we were doing." Most of the scientists were young and few had children, so there was nothing to stop them from working a full day, taking a break for dinner at one of the neighborhood dives, then going back into the lab until 10 p.m. The influx of biotech companies had not yet changed this rundown industrial area, and during Chiron's early years, it wasn't unusual to see empty wine bottles or used syringes in the gutters. In September 1984, Pablo Valenzuela promoted Kathy Steimer from staff to senior scientist. One of her great strengths was her excellent grasp of immunology, a discipline essential for the development of a vaccine to defend against AIDS. Stripped down to its basics, immunology is a study of how the body determines whether something is self, and therefore harmless, or non-self, and potentially dangerous. The basic ability to identify something as foreign is the first step toward a protective response against threats, as minor as a splinter, or as potentially lethal as the AIDS virus. Paul cloned J. Levy's virus and determined its full sequence in September. This was a landmark accomplishment, and Chiron filed for a patent on the genetic sequence of the virus and the general arrangement of its genes. Having staked this claim, Chiron wanted to move fast on products. Kathy was whipshawed from one task to the next, trying to do everything at once, sometimes collaborating with Paul Luciu and sometimes working only with her 5 technicians. Chiron identified 2 genes that coded for parts of the envelope glycoprotein, a protrusion on the surface of the AIDS virus, and on infected cells. Glycoproteins were important antigens in experimental vaccines against feline leukemia virus, influenza in mice, and of course, the successful hepatitis B vaccine being used in humans. Confronted with the AIDS virus, the body's first response would probably be to make antibodies against this knobby glycoprotein. If blood bank technicians could detect these antibodies in donated blood, they would know which units to throw out. Certain parts of the envelope protein elicited stronger immune responses than others, and Kathy performed a series of experiments to pinpoint the most reactive parts. She picked away at genetic sequences to figure out which peptides or small proteins kicked antibody production into high gear. As important as this work was to the company's future, there were other demands on Kathy's time as well. She was working with Paul J. Levy and Dino Dina on the journal article reporting the cloning of the virus. There was a mad dash to get it into print before their competitors did, and on September 22nd, year 1984, they finally shipped it off to Nature, a famous scientific journal edited in London. But there was no time to relax. The Chiron team worked on inserting the glycoprotein genes into yeast, so that the yeast would make the viral proteins when it reproduced. So they're using a bacterial yeast culture and transfecting it so that it makes the protein. Just saying. Actually, Jonathan Cooey, get going biological. Protein production is difficult to measure in cultures of yeast cells, and Kathy's group worked nonstop to accomplish this. During this intensely demanding period, Martin Wilson saw a side of Kathy Steimer that she kept well hidden at work. By that time, they were renting a house together in Benicia, a bucolic waterfront village on the Carquinez Strait, which joins the Sacramento River and San Francisco Bay. Benicia was a geographic compromise for this two-career couple, located halfway between Chiron's Emeryville headquarters and the campus of University of California, Davis, where Martin worked. Kathy struggled with long bouts of depression, during which getting herself off to work was a major exercise in emotional weightlifting. And the longer her work days, the more sleep-deprived she became, and the less able to sleep. And when this happened, she drove herself onward with an unnatural, manic energy until finally Martin would force her to go away with him for a few days, usually to a favorite spot in Baja, California. Photographs from these trips show a handsome couple, his short hair, spiky with seawater, and hers wild and blonde, tanned arms, hugging each other against backdrops obliterated by the sun. In Baja, Kathy stepped off the treadmill. In April of 1984, Secretary of the United States Department of Health and Human Services, Margaret Heckler, told a Washington press conference that a vaccine against AIDS would be ready for testing in 2 years. A former Republican congresswoman from the state of Massachusetts, Heckler had no science expertise and, on this occasion, was suffering with a rotten cold. Minutes before stepping on stage, Heckler asked Bob Gallo, whom she was just about to hail as the discoverer of the virus that causes AIDS, what to say if anyone asked about a preventive vaccine. Gallo pulled a number out of the air. Although this sadly wrong prediction has been attributed both to Heckler's ignorance and to Gallo's legendary arrogance, it may simply have reflected the optimism that ruled early in the biotech era. Researchers in government laboratories, biotech companies, and universities were bursting with ideas about how to make an AIDS vaccine. Jonas Salk was convinced that it would be possible to protect against AIDS with an inactivated version of the virus, an updated version of the paradigm of his successful polio vaccine. Largely on the strength of his name, Jonas Salk quickly raised $20 million to start a biotech company, a move that many investors would later come to regret as stock prices fluctuated wildly. A Harvard primate researcher and a Cambridge biotech company jumped on the live attenuated bandwagon early, even though most experts believed that a live vaccine for AIDS would be too risky. Other startups set out to create AIDS vaccines from synthetic versions of antigenic peptides, short chains of amino acids that some predicted would safely elicit powerful immune responses. Everybody had an idea. Chiron's big score had been the hepatitis B vaccine, a protein subunit expressed in yeast. Ed Penhote said years later, quote, "Clearly the potential for using this technology to make an HIV vaccine seemed relatively straightforward. This is one of those cases where ignorance was bliss." And he said this years later, shortly after he left Chiron to become a university dean. Kathy Steimer's mission was to find out everything that she could about GP120, the envelope glycoprotein that would be, in one form or another, the antigen for Chiron's new vaccine. Early on, Kathy agonized that she would never figure out exactly what this antigen should look like. For months, she worried that some other scientist in some other lab had a surefire way of making a vaccine against AIDS. Gradually, Martin recalls, husband, she came to see that none of the other aspiring vaccine designers and entrepreneurs knew any more than she did. Kathy told Martin, "Nobody knows how to make an AIDS vaccines. We really don't understand the basic immunology that goes with this." In her lab, Kathy studied the blood of AIDS patients in minute detail by carefully analyzing their antibodies against HIV, as the AIDS virus was officially named in 1986. Kathy hoped to determine exactly which part of the virus elicited the strongest immune response. If a vaccine was based on the most powerfully antigenic bits, perhaps it could stimulate a response strong enough to protect people who later encountered HIV in the wild. Kathy missed being able to talk with Paul, whom she greatly respected as a scientist, as she tried to pick the right antigen. But Paul was gone. In mid-1986, when Chiron was becoming less like an academic lab and more like a business, he left the company for a faculty position at University of California, Davis. Paul wasn't really a corporate-type guy, and he had become less interested in making products and more inclined towards laboratory research on HIV and similar primate viruses. His main focus was using animal models to test HIV vaccines, and over the years, he's built a distinguished career and mentored dozens of young researchers. Back at Chiron, Kathy Steimer and Nancy Hagewood worked together to refine GP120. In the lab, they chiseled off epitopes, tiny parts of the antigen, that they thought might interfere with the powerful immune response they were seeking. Because Nancy's special expertise was in expression systems, which harness ordinary cells to make foreign proteins, her role was to make larger quantities of the GP120 pieces Kathy judged most promising. What was that Jonathan Cooey say, if I may? You can't make a cuckoo clock make toast. You can only make something do what it's already capable of doing, what it's already designed to do. So cells can make proteins because Nancy's special expertise was in expression systems, which harness ordinary cells to make foreign proteins. Why would a cell make a foreign protein? It's like hijacking a car factory, the Ford auto factory, and holding the line workers at gunpoint. And by the end of the day, they're going to make BMWs. The factory is designed to make things that the factory is designed to make. Yeah, I'm sure that they could, get out a welder and bend some sheet metal and a little bit of Bondo and stuff. Maybe make a Mustang look like a 3 Series BMW, but That's not really the way it works. Factories are designed to make things that the neat factory is designed to make. So her role was to make large quantities of GP120 pieces that Kathy judged most promising. And I guess the narration here is, why would they expect it to make a good protein if it's not something that the cell would make in any other condition? Maybe it does make it in normal conditions. I don't know. Let's continue on. Just how GP120 should be made was a hotly debated issue at Chiron and elsewhere. The question was whether yeast or animal cells should be used to manufacture GP120, and a growing number of scientists were coming to believe GP120 grown in cells from mammals could more closely resemble the knobby protrusion in HIV. But Chiron's forte was making proteins in yeast cells, so they stuck with that system. And again, what's the immune system do? It identifies foreign versus self. So if you have a protein being injected, it's going to, subtle differences. Was it grown in yeast? Was it grown in rat? Was it grown in a different human? Was it grown in that person? And the immune system may respond, it may or may not, very differently in all four cases. Again, you could hijack the Ford automotive factory and tell them to start making Toyotas. I think when one of those drives down the street, you'll notice that it's a Toyota made at the Ford factory. Once a week, Kathy and Nancy scheduled a working lunch. On pleasant days, they would leave Chiron and walk to one of the new cafes that were cropping up now with... start that one again. On pleasant days, they would leave Chiron and walk to one of the new cafes that were cropping up now that scruffy Emeryville was beginning to gentrify. Both were blonde and blue-eyed. Nancy was 3 years younger, a tad shorter, and prettier in a more conventional way. Both were being promoted up the corporate ladder in the late 1980s, and had traded jeans for a softer, more corporate look. Nancy was ambitious and self-assured, and for the most part, she shrugged off adversity and criticism. So, this is interesting. You have two blonde, blue-eyed women, almost the same age, in competition. I don't mean to spill the beans here, but before this book ends, Kathy Steimer is dead. Nancy Hagwood is still alive, and several years after this book was written, Nancy Hagwood was the person that allegedly went to the FBI and said that Bruce Ivins was responsible for the anthrax letters. And as this book shows, Paul Lushu was one of the most frequent co-authors of Robert Malone's postdoc, Murray Gardner. Nancy Hagwood is the number 2 or number 3 most frequent co-author of Robert Malone's postdoc, Murray Gardner. It mentions Robert Malone. Robert Malone had been working in the laboratory of Bob Gallo just before September 11th, 2001. And all the people that Nancy Hagwood worked with, including people like John Wolfe and Gary Rhodes, are also the co-authors on Robert Malone's paper in 1989, meaning Robert Malone is at the very least, connected with, on some friendly level, I don't know how close, with Nancy Hagwood, the person that said, went to the FBI, allegedly, said that Bruce Ivins did the anthrax letters as far back as 1989. That is quite a thing. That is why this book has a lot of buried stuff in it. Now, was Kathy Steimer killed? I don't know. I mean, the book suggests that she just worked too hard and died of exhaustion, gave herself cancer, or something like that. Her husband and her adopted daughter still work at University of California, Davis. Stakes are very high here, folks. Between Kathy Steimer and Nancy Haygood both were blonde and blue-eyed. Nancy was 3 years younger, a tad shorter, and prettier in a more conventional way. Both were being promoted up the corporate ladder in the late 1980s and had traded jeans for a softer, more corporate look. Nancy was ambitious and self-assured. For the most part, she shrugged off adversity or criticism. Although she tried to hide it at work, Kathy Steimer was by far the more sensitive of the two. When Kathy traveled to conferences at Cold Spring Harbor Laboratory in Long Island and other temples of the East Coast scientific establishment, she was surrounded by men who never lost sleep wondering whether they were wrong about anything. When she rose to question an experimental procedure, she was often dismissed and sometimes even attacked. Kathy told her close friend Catherine Radke, R-A-D-K-E, also a retrovirologist who knew the major players, that she didn't know whether she could ever go to another AIDS conference, quote, where all the big egos and high rollers were jockeying with each other for glory, unquote. This may have been after the meeting where a famous Duke researcher loudly propositioned Kathy while his inebriated dinner companions egged him on. As upsetting as these East Coast meetings were to Kathy Steimer, life was good in California. At Chiron, Kathy Steimer was beginning to bring in outside funding for AIDS vaccine research. She started small in 1987 with a $500,000 National Institutes of Health (NIH) grant to work on genetically engineered vaccines. In 1988, Kathy Steimer and Nancy Haygood were co-principal investigators on a $3.1 million AIDS vaccine grant from the state of California. Kathy soon got an additional $1.3 million from the NIH on her own. Chiron had also begun manufacturing GP120 and mammal cells instead of yeast. Lunching in the California sunshine, talking about the temporary setbacks and ego battles that were inevitable in any kind of work, Nancy and Kathy never doubted that genetic engineering would enable science to win the war against AIDS. With the recombinant hepatitis B vaccine as a precedent, how could they fail? Nancy Haygood later recalled, quote, "There were so many ways that we could make this vaccine. It was just a matter of who was going to get there first," unquote. Sure, there were difficulties along the way, but they were going to prove that they could beat the virus. Kathy's personal life was also on the upswing. She and Martin had been together since 1982, and in 1987, they moved from their rental house in Benicia to a quirky, dramatic home designed just for them. On the outside, it looked like a modest frame house onto which a boxy two-story concrete addition had been grafted. Inside, the space was airy and coherent. The site commanded a spectacular view of the Carquinez Strait to the hills of Port Costa, a small town on the opposite shore. Kathy and Martin gardened with a passion, planting great rosemary hedges and fruit trees with a special fenced enclosure for Leroy, who was their black standard poodle pup. Photos from that time show the two of them with a dog as dark as an ink blot squeezed in the middle. With the house and dog in place, the absence of the child they had been trying to conceive for years became acute. Finally, they began to investigate adoption, only to discover that the agencies favored couples who were married, which they weren't. They simply hadn't felt the need to make it official. Each had been married before, in Kathy's case, a graduate school misjudgment that had lasted less than 1 year. Martin's first marriage had lasted longer and had a more amiable parting. But with Kathy, she had not felt that a wedding was needed to cement their bond. Now, it appeared that if Kathy and Martin wanted a child, which they did, they would have to tie the knot. So in December of 1989, they went to Tijuana with Kathy's father, Harry Steimer, and his lady friend, Sandy Davis. Quote, We paid a guy $5 to make sure that the hubcaps didn't get stolen off the car while we went into one of those places with a big sign out front that said marriages and divorces. It seemed like it would be much more fun than having to have a big production. This was recalled by Martin. Afterward, the foursome went out for margaritas. Harry and Sandy took the Tijuana trolley back to San Diego, while Kathy and Martin took a road trip to Baja, California. They returned from their honeymoon to find an answering machine message from a young woman who thought they sounded like a good adoptive parents for the baby she was carrying. Kathy's date book for early 1990s was crammed with appointments with lawyers, doctors, social workers, all the people who became involved when an adoption is being arranged. On March 25th, Kathy and Martin were at the hospital when the baby they named Acacia Lynn was born. She went home with them to Benicia the following day. Kathy took a month off from Chiron, although now and then she went to Emeryville for a day, jammed with meetings. Both she and Martin worked part-time for 6 months, and Casey, as the baby soon came to be called, was the center of their lives. For the first time, Kathy stayed up late with an infant instead of an experiment. Except for the addition of the baby, their overall domestic arrangements were the same as ever. Kathy handled all the financial accounts, and Martin, an expert and talented chef, did all the shopping and all the cooking. Meanwhile, Nancy Hagewood, who had been made co-leader of the HIV vaccine project and associate director of virology in the early 1990s, ran the show while Kathy Steimer was on the leave. By this point, Nancy Hagewood was using mammalian cells to make GMP lots, good, large batches of vaccine that met federal standards for good manufacturing practices, GMP. Before the United States Food and Drug Administration will agree to let a vaccine be tested in humans, it demands that the safety of the product has been demonstrated in animals, that the identity and purity of the product can be assured, and that the product will be stable during the course of a clinical trial. During the summer of 1990, Nancy and other Chiron representatives met with NIH officials in Bethesda to talk about a government-supported trial for GP120. The prospects looked good, and Chiron submitted an investigational new drug IND application for its recombinant GP120 vaccine to the Food and Drug Administration. The IND described in great detail all the cutting-edge molecular biology, all the novel manufacturing techniques that had gone into the making of this new vaccine. As dazzling as all this high-tech wizardry was, there was still only one way to prove whether it would actually protect people against AIDS. Immunize volunteers and see what happens.